Now, please let me introduce the second speaker, Dr. Ming Ni. Dr. Ni graduated from the University of Washington in the United States. In January 2020, he joined GenScript and assumed the responsibility for technical support of the plasmid DNA platform. He led the plasmid DNA project, enhancing upstream fermentation yields and optimizing downstream purification steps, managing multiple CMC projects for both plasmids and viral vectors. He achieved 4 R&D approvals, including FDA, NMPA, and CCMO. Additionally, 2 projects are advancing to the clinical phase 1A stage. In this talk, Dr. Ni will discuss accelerating the development of the cell therapies via manufacturing high-quality plasmids and high-titer viral vectors. Now, I'm delighted to hand over to today's speaker, Dr. Ni.
Thank you very much for your kind introduction. Yeah. Can everyone see my screen?
Yeah, we can see. You can play the PowerPoint.
Yeah. Okay.
Okay, that's great.
Thank you everyone. Good afternoon. Today I'm very delighted to share with you in this webinar entitled Accelerating the Development of the Cell Therapies while Manufacturing High Quality Plasmid and Viral Vectors. As many of you know, cell therapies, including many different types of therapies, for example, like a bone marrow transplantation, stem cell therapies, but most recently, CAR-T cell therapy really achieved success in treating cancers, moving from blood cancers now to the solid tumors. In this recent published review paper, you can see that the key milestones for the CAR-T cell therapy dating back to more than 30 years ago in 1987, actually a Japanese immunologist, Yoshikazu Kurisawa, he actually introduced the concept of CAR-T and using the genetically engineered T cells to trade animals and later achieved success. Fast forward to 2017, the first commercial product, Kymriah, got approval from FDA to treat leukemia. So this is really a great success to commercialize the cell therapy. And so far, FDA already approved 6 commercial CAR-T therapies. Worldwide, totally 12 CAR-T therapies have been approved, including 5 from China and 1 recently approved in India. So this is really important to make CAR-T cell therapy really available for patients to provide high quality plasmid and viral vectors to make this thing happen. So in today's webinar, I will divide my talk into 3 topics. The first one is how to enhance the quality of plasmid for CAR-T cell therapy manufacturing. The plasmid DNA, they are the crucial raw materials for the cell therapy to make really the CAR-T works. And secondly, once we get the plasmid, now we move into the viral vector part. We need to maximize antiviral titers for CAR-T therapies. Last but not least, I was talking about at GenScript ProBio, we provide you FDA, EMA ready CMC. With that, can really accelerating the antiviral vector development cycle to shorten that from 1 year to only about 5 months. So first, let's look at how we are going to enhance the quality of plasmid DNA for the CAR-T cell therapy. So as you all know, for the lentiviral system, you have different generations. GenScript ProBio are using third generation lentiviral system, which including 3 helper plasmid and 1 the GOI. That is quite important. So it's 4 plasmid system. This is third generation. And our platform, which can provide you a high quality plasmid, we are using high density fermentation. And we are also using the chromatographic purification to make our product really guarantee you to have high quality. So first, let's look at the process. Over the years, at GenScript ProBio, we established robust circular plasmid manufacturing process, starting from the plasmid construction all the way to final fill and finish to have the product. The key point I want to emphasize here is our fermentation part. First thing, we have the E. coli strain. Those E. coli strain are IP clean, and we also hold sub-license right. You can use it, it's trouble-free. And secondly, the whole process is antibiotic-free and TSE, BSE-free. Last but not least, we are using high-density fermentation. We can provide cool titer around 1 gram per liter, which is really impressive. For the downstream purifications, we are using 3-step chromatographic purification steps, including gel filtration, affinity chromatography, ionic exchange chromatography. We are using 3 steps chromatographic purification because this can guarantee the high quality. And very proudly, we can guarantee you to have a pretty high recovery rate between 30 to 60%. This is a really good number in comparison with our competitors. First, let's look at the E. coli strain. E. coli, those are the working horses for producing plasmid DNA. Of course, now you have a cell-free system competing with E. coli, but traditionally people still focusing using E. coli, and also this can provide you at this stage. Still, they are the working horse, can provide you large-scale, high-quality plasmid. So for our E. coli strain, very importantly, those E. coli strain, they are IP clean, and we host the sub-license right. You can use it without any trouble. And very proudly for, as I mentioned, we use a third generation lentiviral system, which including 3 lenti-hyperplasmid, showing here. Yeah. So those lenti-hyperplasmid, we are already registered with FDA. We get the SEBA FDA registration number, and this really can shorten your development cycle to push your CAR-T product to the early clinical stage and later for commercialization. Those are very important, and those FDA DMF register number really can shorten the development period. Next, we need to talk about the cell banking. Very importantly, the E. coli strain, although they are IP-free, IP clean, and suitable for CDMO uses, very importantly, we need to do the screening process to screen the right cell banks later for encouraging later we can use it for large-scale production. So very importantly, we need to screen for the strains with good passage stability. So here gives you 2 different strains. One has a bad passage stability, and the other one has a good passage stability. So how we determine that? You can see for different passage, we do the AGE gels. You can see the bands. If this string has good passage stability, as you can see, only one band can be C. This is really good. If it has bad passage stability and through the passage number increasing, you can see visually, you can see the second band. Although at passage 8, here you can see very weakly those second band, but very clearly at the passage 10, this second band can be clearly seen. And another important thing is looking at the plasmid yield over the passage number. As you can see, although string A gave pretty high number at the early passages, and this number is decreasing over the passage number increasing, and while the other one, although the number is not that high in the beginning, but is quite stable over the from passage 1 to passage 10. So this passage stability really can guarantee later you use for large scale production, you don't have any problem with your plasmid DNA product. So take home message, screening a stable string with high plasmid retaining rate is very important. With this string, this can be suitable for the large scale production for later commercialization and different clinical stage use. This will be good. So next slide, I want to talk about our upstream process optimization. We use high density fermentation, and the key point is after optimization, we optimize different parameters. For example, for the culture media, we can optimize the pH, the components for the culture media, and also the concentration. And when we fully optimize all these parameters, we really can improve the yield over the time. As you can see, for one case study showing here, we can really increase the fermentation yield about 3 times. And we can really achieve high yield fermentation up to 1 gram per liter. This is quite impressive. So next slide, talking about the downstream process. Well, we not only optimize the fermentation steps, very importantly, the downstream purification need to be optimized because 2 important indicators for purification steps. One is to guarantee get high recovery rate, stable recovery rate, and also at the same time, we should maintain the impurity level to be low. And of course, to check the quality of plasmid DNA, we look at the supercoin content. In this case, IEC content percentage. You can see that after optimization, we can achieve a pretty good recovery rate. The recovery rate can be increased from 2 times up to 3 times. And on the other hand, we can maintain the impurity level at a very low stage. And also for the supercoin content, we can get it above 90% easily, sometimes even close to 100%, 99, 98%. So this is the way we really can achieve high quality plasmid DNA. This can ensure you later for the lentivirus production, you have high quality plasmid, then to achieve the lentiviral production success with this high quality plasmid will be very important. So with that, I hope I already convinced you we can achieve high quality plasmid. Now we move to the lentiviral part, the production of lentiviral vectors. So for the lentiviral vector platform, we have two different systems. One is the adhering culture system, and the other is suspension culture system. At the GenScript ProBio, we have both systems for our clients to choose. First, let's look at the adhering culture system. So here shows the adhering process. This is showing the flow chart, starting from the cells. You saw in the cells, you expand the cells, and all the way to the lentiviral virus packaging, and later, very importantly, to do the purification steps. And last but not least, the quality control of the product. For our lentiviral adhering platform, upstream, we are using HEC293 T cells. Those are being authorized for CDMO use. And also those cells also get the DMF registered number, so you can directly use for the later IND submission. This is quite good. And at this stage, we can maximize the scale at about 48 liters up to 54 liters. Later, I can share with you with the real data. And for the upstream, when we harvest the virus, the harvest titer is around 10 to the power 6 to 10 to the power 7 TU per mil. This is a quite decent number. And for downstream, we can get the final product to achieve the lentivirus letter above 5 to the power 7 TU per mil. And for the recovery rate, depends on different projects, depending on the molecules, we can achieve a recovery rate between 30 to 50%. So very importantly for the GMP manufacturing is to show your process is stable and also you achieve a high recovery rate. Here showing the example in the case one, you can see when we culture cells in the cell factory, this showing different cell factory number. Over the time, if we increase the cell factory number, we can achieve quite stable the lentivirus titer. You see the variation even from 1 up to 9. The variation is quite small. It's only about 7.2%. This show that we can get quite stable process. And on the other hand, in terms of the recovery rate, they are quite high. In this case, we can get close to 60%. This is good number. And also in terms of the impurity, for example, the endotoxin level, the number is really low. And another important case is showing the process can be scaled up. So here we show that scaling up from 4 liter to 24 liter, and later in the GMP workshop, we do it 48 liter. Over the time, you can see in terms of the final product titer, they are quite similar. And also in terms of the recovery rate, they are also quite similar. This showing that our process is stable and also can be scaled up all the way to 48 liters. And in terms of the recovery rate, we still can get quite good recovery rate. So this case study is showing that how we optimize the downstream purification. So for downstream purification, actually, lentivirus production downstream is more complicated compared to the plasmid DNA. So there are many parameters you need to tune. And as GenScript ProBio, through the years, we really find out some very important parameters. By tuning those numbers, we can optimize the downstream purification process to achieve a recovery rate. Here we show 3 different cases. We show that actually the recovery rate can be increased from two times up to like 4.5 times. And recovery rate now has been improved. Also, at the same time, very importantly, we should keep the impurity at a very low level. Here shows, for example, BSA level, the host cell protein, host cell DNA, those impurity level are quite low. And in terms of the plasmid, the residue of the plasmid, you can see that is still at the low level, which can guarantee the lentiviral product with high purity, with high titer can be used for clinical trial later on. Another case is we want to show that in case of scaling up from 4 liter to 24 liter all the way to 48 liter, we can do that quite confidently to achieve quite similar P24 and the FAX titer. And in terms of the impurity like HCP, HCD, and plasmid DNA residue, they are keeping at a very low level. And if you look at the summary for the downstream at different scales, you can see the final titer quite similar. And in terms of the impurity, also those numbers are quite similar. We can meet the requirement of our clients, and also those are quite good for filling up for IND approval. So far, we have achieved many cases to achieve the success with those high purity lentiviral product. Another important thing is at the GMP workshop, you do it at a relatively large scales, like scales above 48 liters. Very importantly, you should be confident and comfortable to do that at a relatively large scale under the GMP conditions to achieve, still achieve high high titers, and also to improve the product with low impurity, and very importantly to meet the requirement for IND approval for the IND requirements. So with that, I hope you get convinced that we can do it using our adhesion process to achieve large-scale GMP production, to get high titers and with relatively high recovery rate and also low impurity. So next, I will talking about our lentiviral vector system using suspension culture system. Suspension culture is the future for lentiviral production since it has many benefits, such as scaling up, and also they're using serum-free system. So this is really the future for the lentiviral production. So here I want to share with you the suspension process, showing in these slides the flow chart of suspension process. And you can see that it's also starting from cell recovery. First, you expand the cell in the shake flask, and later you move to wave bioreactors. And later, when you get enough cell numbers, you put into the bioreactors, like 50 liter or even more, to do the lentiviral production. And the key point I want to emphasize is first, for the suspension process, we are proudly to provide you the PowerS 293T cells. Those cells we have own IP, and those cells also get DMF registered. And for the upstream, using the bioreactor, now we can do at 200 liters, which is quite decent scale for production. And for the harvest titer, we can get it from 5 times to the power 10 to the power 6 up to 1 times to 10 to the power 8 TU per mil. And the final product, we can guarantee to give you the titer above 5 times 10 to the power 7 TU per mil. And for the recovery rate, we can get it between 30 to 50%. So first, I want to spend a minute to talking about our cells. We have PowerS 293T cells, which is the excellent choice for suspension culture. We have own IP, and those already being registered with DMF number. And those cells are compliant with NMPA, FDA, EMA, all these regulatory agency. And you can see from both the optical and the fluorescence microscope, you can see those cells very well distributed and with low aggregation rate. And we use this cell to package different molecules and the lentiviral production. For that, we can achieve high transfection efficiency, and which guarantee later to get a pretty high yield. And also for the cells, very importantly, during the cell culture, those cells still maintain high viability, and those are also suitable for high density cell cultures. And very importantly, compared to commercial available other cell lines, the doubling time of our PowerS 293 T cells, those doubling time is a little bit shorter. And those for the packaging of lentivirals here so that we can easily get the final titer above 1 times 10 to the power 8 TU per mil. And we have used it for variable projects. And down to the bottom right, you can see that those we have the patents, and the cell has been reserved by the Chinese cell banking system. So with that, I want to talk about the process. Yeah, for process, very importantly, you should have a very stable process. We have tried, different projects, different molecules. As you can see, the harvest titer, ranging from 2 to the power 7 all the way to 1 to the power 8. And the recovery rate, the range is between 40% to 50%. In terms of the impurity, HCD, HCP, plasmid residue, they are relatively low. So we have done various projects, and we have demonstrated that with our system, the process is quite stable. And next, very importantly, is the scalability. We have done that to showcase from 2 liter, 10 liter, all the way to 50 liter. In terms of the titer, you can see the number is quite similar. In terms of the cell performance, you can see the cell density, the cell viability at the different scales like 2 liter, 10 liter, 50 liter. Their performance is quite similar. And in terms of the product, the final yield, they are in the same range, around 2 to 3 to the power 9 TU per liter. And the recovery rate, depending on the project, so somewhat is around 20%. And also in terms of the impurity, the number is quite similar, and we can get quite low impurity for those lentivirals. So the last case, very importantly, for suspension culture, people always question whether they are similar for the lentivirals produced by the adherent process. Here we show that using the T cell, we can show that first, in terms of the titer, when you use a suspension culture system, we can get pretty higher lentivirus titer in comparison with the adherent process. This is the first advantage. You can see for different projects, we can achieve from 2 times all the way to 15 times. That's a very important for later scaling up. So at the same scale, using suspension culture can give you more lentiviral product, which can supply for more patients. This is quite important. And not only it can give you the titer is higher in terms of the T cell transfection efficiency. We have done head-to-head comparison, compare the suspension system and adherent system. And you can see that in terms of the CAR positive cells, the CAR positive rate is about 70%. And it shows that both suspension system and adherent system, they perform quite similar. This is good. And in terms of the cell we are using, once again, it shows that consistent. You can keep the cell expansion in the large scales, no problem. And also in terms of the cell viability, they still keep a pretty high cell viability. So with that, I hope I convince you our suspension system is quite good in terms of the process is stable, can be easily scaling up. And compared to adhering system, it can even provide you more lentivirals in terms of the T cell performance. It also can give you quite similar T cell transfection efficiency. So in the last part of my presentation is to provide you FDA EMA ready CMC. The key point here is we can help you to achieve the IND approval, both in FDA and EMA. But at the same time, for the CMC project, we can complete that with much shorter time, using much shorter time. So for the FDA EMA IND ready lentiviral vector CMC, typically it can be done within a year, 12 months, starting with cell banking, plasmid production, and optimization of both upstream, downstream, and later for the lentivirals, you also need to optimize upstream, downstream process, and later pilot study. And the total process will take more than a year if you include the stability study. For our FDA EMA ready CMC, we can do that within 5 months. So the thing is, actually, we start with cell banking, and later we use our platform technology, and we can directly do the pilot scale production, and then later for the stability study. With that, we can get the IND documentation ready, and for your project, the CMC, the cycle is only 5 months. This is really good news for the startup biotech companies, and for those companies with a little bit shorter in terms of the funding, and also quite important to push the product to the clinical study. This is quite important. Now I want to spend a minute just talking about our plasmid and viral vector service to give you the overview. Actually, we have 3 different grade of the product for both plasmid and antivirals. We have pre-CMC grade, we have a GM Pro grade, and also for the GMP grade. Depending on which stage you're at, for the research and discovery stage, you can use pre-CMC grade without any problem. And later for the early stage clinical trial, like phase 1 or phase 2, you can use the GM Pro and the GMP grade product. And for the late stage and commercialization, definitely you should use GMP grade. And also depending on the applications, for example, you are doing plasmid, not for just limited to CAR T cell therapy. For example, you're doing DNA vaccines, those are considered DP, then you should, of course, use GMP grade for clinical studies and later commercialization. And similarly for viral vectors, you should use GMP grade for phase 1, phase 2, phase 3 clinical studies and later commercialization. At the end, I want to wrap up my presentation and to give you an overview of GenScript ProBio. Over the years, we are quite young in terms of the history of this new company, GenScript ProBio. We are the branch of the GenScript, which you're probably familiar with. Over the past 3-4 years, we already achieved excellent track record, and you can see that we can get over 30 IND approval globally. This number is still increasing, and currently we have over 60 IND projects are ongoing, and we are proud to see that we have completed over 300 GM Pro batches. And here showcase for different projects, for different applications, we already get IND approval. So this list is keep ongoing and ongoing. And the last slide is our commitment. GenScript ProBio is dedicated to customer first. We ensure the quality. We provide you the innovative solutions, and we also provide you very flexible business models. We hope you will become our service can become your most reliable and sustainable biologic partners. With that, I will end my presentation. Thank you very much for your attention. Yeah, now I hand to Jessica.
Thank you for Dr. Ni's excellent presentation. Let's come to the Q&A part. Yeah, okay, we received some questions. Let's look at the first question. How to achieve the crude titer of plasmid DNA up to 1 gram per liter?
Yeah, this is one of the the key points we want to highlight. Not all our competitors can really achieve such high plasmid DNA production, and we have to say for not for every project we are working on, we can get such high plasmid crude titer, but for certain plasmid molecules, we really can achieve such high plasmid DNA production, the titer, through 3 different strategies or approaches. First, we use the E. coli strain, which I have shown that it has stable passage stability. This is first thing. And secondly, for the upstream process, we optimize key parameters. We are able to get pretty high crude titer. And of course, there are many know-hows, which, for example, the carbon source, the nitrogen source, how we manipulate those parameters. We are, in the end, in short, we can really guarantee to get such high crude titer. Yeah, thank you for your question.
Okay, thank you for your answer. The next question is, as the timeline for LVV-CMC takes about 5 months, how long does stability test take?
Oh yeah, that's a very good question. But for stability, we have to do that according to the pharmacopoeia. We do it, typically the stability is also associated with your product. How much longer can you still use it? The shelf life of your product, for example, antiviruses, you can use it for 2 years. Then for stability study, you have to do it for 2 years. Yeah, this is this is important. But what I'm saying is, for the FDA and EMA ready CMC, excluded the stability. We do that in a relatively short time, because typically CMC project involving upstream, downstream optimization process. Then we do cell banking, and then we directly use our platform technology to do the pilot study. With that, we can shorten the CMC period. Yeah, but you have a very valid point. We have to see that stability. We have to do it to guarantee that 2 years later you still can use that antivirus we produced for you. Yeah, good question. Thank you.
Okay, thank you. And the next question is, do you have both the adherence system and suspension system in order to customize according to the method the customer wants? Or do you also recommend a method that can show better efficiency? Is the basis for recommendation founded on existing know-how?
So first, for both hearing and suspension process I showed today, we have we call it the platform technology we can directly use. And this know-how and the whole parameters we established through our R&D process, through our multiple CMC projects. We keep improving that. And this platform technology can really help our customers to shorten the period for CMC. And for you, we can customize. First thing is, we can customize the process for you. Yeah, because for cell and gene therapy, each application or each indication is unique, and your solution is also unique. Then how one can just use one set of parameter suit for your projects, right? Then if you look at each individual CMC project, we really customize the parameters, the process, and this parameter, this process belong to our customer. Over the time, we developed that, but this is for you for the submission, and all the documents, all the properties, products belong to you, our customer. And definitely we will customize the process to suit the needs for our clients. I hope that answers your question. Of course, if you have any follow-up question, I'll be happy to answer that. Yeah, thank you.
Okay, thank you. The next question is, for all the commercial CAR-T products, adherent process was used. Why?
Yeah, the adhering process is used. This is the fact. And of course, as I said in the presentation, suspension is the future. It definitely have many benefits, but adhering process, for all the commercial product, of course, this has a history. And also using new technology, using suspension, the system, you need to do a lot more to prove that the final lentiviral product is similar, equivalent, or even better compared you use adhering process. This will take some time. Eventually, we can achieve that, but at the current stage, the commercial products still using adhering process. I hope in the near future we can use suspension system. Yeah, thank you.
Okay, thank you. Yeah, the next question is looking at recent articles, it seems that the tension between the US and China is intensifying. If production is done in Chinese manufacturing plants, will it become more difficult to pass FDA inspections? I'm curious about the audience opinion. I hope
Yeah.
I'm just worrying unnecessarily.
No. I think this is a really fair question. From myself personally, last October, December, my projects, we have visitors, our clients from European countries and from United States come to us. They work with us for up to three years. They also talking about the geopolitical threat. You are right. Currently, the tension between two superpowers. First, I have to say that I'm Singaporean. Singapore is is a country like Switzerland, so I don't take sides, right? So those two superpowers, the tension between two countries getting, the situation, I would say, getting worse compared to 1 year ago or compared to 2 years ago. And for that, I have to say that, GenScript ProBio, we already think about it. Actually, we are proud to say we are a US company. We have a manufacturing site based in New Jersey, and we will open our plasmid facility, open for service in December this year. And our headquarters for GenScript ProBio is based in the United States. So we are American company. So for that, I hope, I don't know whether you're from Korea or other countries. I hope my answer satisfies you and you can work with us and to, circumvent this problem, geopolitical problem. Yeah. Thank you.
Yeah, thank you. And the next question, I think it's already been described during your presenting. It's what are the benefits for choosing suspension process over adherent process?
Yeah, adherent process, as you already mentioned, those are currently used for commercial products, but the suspension is the future. One, it holds, you can see from my slides, suspension process really gave much higher titers in terms of antiviral production. Sometimes you can even achieve 10 times, 15 times higher. This is the benefits. And for the system, it also holds the promise for scale up to large scales. For the adherent process, we use cell factory. This has limitation for scaling up. As I said in the presentation, now we have 48 liter, 54 liter, but compared to suspension system, we can easily scale up to 200 liter. Yeah, this is one of the advantages. And also for adherent process, you use BSA, you use serum, because serum contains important factors for cell to be able to adhere to the cell culture substrate. But the suspension, you can use serum-free culture media. You don't need to worry about the BSA. You don't need to worry about the serum. This is a lot better. And of course, suspension system still facing many challenges. We have to deal with that. Yeah, for example, how they perform in the T cell performance. Yeah, adhering and suspension system give you lentivirus, and you use it because you think about how they being packaged. Adhering is more or less like a 2D system, right? Suspension culture cell in the 3D system, and then the packaging process at the molecular level is very different. And there are many challenges we are facing. We need to solve the problem to really guarantee not only we can get higher titers, but later we get high quality lentivirals similar to that adhering process, or even better than the adhering process. That's the bright future for the suspension system. But we need to overcome some problems on the way. Yeah. Thank you.
Yeah, thank you for your sharing. That's all. Oh, okay. I see another question.
Yes,
do
please.
Do you also assist with vector design for plasmid DNA? How do you handle it if a client requests this service?
Yes, the short answer is yes, we can help. We can assist our clients to design the plasmid DNA. This is actually our benefits. We can help our clients. Yeah. So if you're interested, you can, later contact me or my clients, or my colleagues, and we can help you solve this problem offline. Yeah. So we have the capability to do that. Yeah.
Okay. Yeah. That's all the questions by now.
Thank you very much.
Yeah. Thank you. Thank you all. That's all we have for today.
Yeah.
Yeah. And also, we are going to hold the offline symposium about CGT on 23rd May and biologics on 5th July. If you are interested in it, please kindly register your interest through the link in the chat room. If you have any more questions, please also contact us through our official email. Let me join others in thanking Dr. Chen and Dr. Ni for their time today. Thank you all for attending today's webinar. Look forward to meeting you again soon.
Thank you. Have a good one.
Thank you.